emergency room case, and it's neuro-related. And we're going to learn a little bit of neuroanatomy, although you guys know me by now and you know that I'm not a really super awesome anatomist, but nevertheless, we're going to learn a little bit of neuroanatomy, and we're going to try and essentially diagnose a particular emergency room case, okay? So if you have a chance to go back to your rooms, I know some of you commute, but if you have a chance to bring your textbooks, I'll bring mine in as well to use as a reference. And you can use computers as a reference as well. So again, you're going to be sort of your own investigators looking into this particular case, okay? So we're finishing up this idea of how action potentials propagate down axons. And we now are experts on the mechanism of an action potential, right? We know that the voltage-gated sodium and potassium channels are instrumental in the steps involved in an action potential. We're going to be spending a little bit of time talking about the velocity of action potentials. And it's really important to remember that the magnitude of action potentials in a neuron doesn't change, right? It's all or nothing. You always get the same magnitude of voltage change of an action potential. However, the frequency of action potentials down an axon is going to determine the strength of the response, right? And so it's going to be important to talk about how the action potential propagates down the axon, and we're going to talk about myelination, and that this myelin sheath that's around axons is going to improve the velocity of an action potential. So recall that we talked about on Friday that the velocity of an action potential down an axon depends on two things. The diameter of the fiber, and just remember the bigger the diameter of the fiber, the faster the velocity, just like a cable, an electrical cable. And then the insulation, which gets into myelination. So the more insulated the axon, the higher the conduction velocity can be. So nerves that conduct the most rapidly, like 100 meters, 100 meters, like the 100-meter dash, right? Everyone knows what 100 meters is about, right? I'm terrible at approximating distances, but at least I can approximate track distances. So 100 meters, right? 100 meters per second. So neurons that conduct the most rapidly are involved in mediating your super rapid reflexes. Things that are regulating your posture, for example. Things that are regulating your spinal reflexes, where you don't even need integration of the brain, right? So things like removing your hand from a hot surface, right? So those are going to be your fastest conduction velocities, 100 meters per second. The nerves that are involved in slower conduction are obviously not going to be those that are going to be necessary for reflexes or for fight or flight responses. So these are going to be more your housekeeping activities, so more your parasympathetic neurons, right? Your neurons that mediate functions of the parasympathetic nervous system. Secretion of things from glands. Visceral tone, that means the tone that's involved in your vasculature, right? You're never all vasoconstricted or all vasodilated. You have a certain amount of tone, and that's going to be mediated by your parasympathetics. So those neurons don't need as fast of a conduction velocity. So let's talk a little bit about myelination. So that's going to play a role in both the diameter and the insulative properties of axons. So your larger nerve fibers, so a nerve, by the way, is just a bundle of neurons, right? So one neuron is a single cell, but if you have a bundle of them that are going to be activating, for example, a set of muscle fibers or a particular set of cells, that's called a nerve. So larger nerve fibers are myelinated. So in the central nervous system, the myelinated, the cells that make up myelin are called oligodendrocytes. Those are glial cells. And in the peripheral neurons, those cells that myelinate are called Schwann cells. So again, both of these different types of cells are glial cells. Those are not neurons. Those are the cells that are like the sheets of cookie dough that I talked about on Friday, that you roll them out, and then if you wrap them around the axon, they're providing that myelination. So again, if an axon was coming out of the board at you, right, the axon is coming this way, this would be the neuron. Ooh, that's not a good sound. There we go. And so that's the neuron. That's the one that's going to conduct the action potential. But then if you had this roll of glial cells, either oligodendrocytes or or Schwann cells, depending on whether you're in the central nervous system or peripheral. If you rolled those cells into a sheath and then you actually formed kind of concentric rings of that layering of glial cells around the axon, it's all these cells that are myelin. And so just like a rubber insulated material on a wire, these are going to insulate the neurons. So a couple things are accomplished here. We now know that action potential velocity is mediated by two things, diameter and insulation, right? So myelin is going to provide both, because aren't you increasing the diameter of the neuron, right? You're increasing the diameter by myelinating, right? You're going from this diameter to this diameter, and you're also obviously improving insulation. So this is all physics. This is all based on decreasing membrane resistance. So if you guys remember your circuits, I know some of you haven't had physics yet. As science majors, hopefully you have physics at least in high school. So in any circuit, resistance is going to be a bad thing, right? So that's going to reduce efficiency of electrical conductivity. So resistance is going to resist flow of current. So if you reduce the resistance, that's going to increase your membrane or your action potential velocity. So membrane resistance is decreased when your fiber diameter increases, right? And when your insulation increases. And so we can basically define this as this resistance equation, where our little r sub m is the total resistance of the membrane, and that is equivalent to the specific membrane resistance, meaning the resistance in one portion of the membrane, divided by 2 times pi times the axon radius. So, yes, you do need to memorize this equation, but this can be essentially reduced to thinking about two things. First of all, the total membrane resistance is going to be lower if you increase your axon radius, right? So this is the denominator. So if I make my axon radius bigger, that's going to make my total resistance smaller, right? It's just a numerator-denominator thing. So the axon radius is essential to understanding action potential velocity. So the less leakage of current, the bigger the axon, the less leakage of current, and local current can be carried longer distances if you have more insulation and bigger axons in terms of diameter. So myelination contributes to increasing axon diameter and insulation, and this brings us to a phenomenon called saltatory conduction. So again, you guys can predict that something like this is probably going to be on the exam in terms of a definition, okay? So let's just take this line by line. So no action potentials actually occur on a myelinated membrane. So let me just sketch this on the board. So here I have my dendrites, right, my cell body, and here I have the axon, and here I have the axon terminal. So you have to imagine that this axon has been myelinated, right? It's got layers of glial cells that are surrounding the axon, but they're going to be in patches. So think of these as kind of different sections of carpet or rug that's going to be surrounding different patches of the axon, right? And if I were to take any one of these patches and turn it so that the axon is pointed out of the board at you, you'd be looking at it like this, right? Where the neuron is here and the myelin is wrapped around it, okay? So what you see here is that there are these kind of naked sections of the membrane, and it's the naked sections of the membrane, the unmyelinated patches of the membrane, where the sodium-potassium channels are. The voltage-gated sodium and voltage-gated potassium channels are. The myelinated sections actually don't even have voltage-gated sodium and voltage-gated potassium channels. So action potentials don't even happen underneath myelin. They don't have to. So the myelin is causing this insulative property such that, okay, if I conduct an action potential here, and I have that wave of depolarization, this section of the axon is so well insulated next naked part of the membrane. And that's where you have sodium and potassium channels, and you're going to elicit more action potentials. And then again, you're going to have this extremely fast spread of current under the myelinated portions of the axon. This is going to help this velocity of action potential propagation incredibly, right? Because imagine that if you have an axon and it's completely a naked membrane of axon, it doesn't have any myelin, every single patch of that membrane is going to have to conduct an action potential, right? Versus if you have this wicked long axon and you've got sections of myelin, and you don't need to have action potentials in those sections that are coated with myelin. So what's happening is you actually are having action potentials that are essentially jumping from naked section to naked section to naked section. So if I have an axon that's a foot long, right, that action potential is going to be able to get to the axon terminal way faster than if I had no myelin. Because the action potential, you can literally think of it's just skipping. And these are called nodes of Ranvier. Ranvier was just the scientist who identified these under the microscope. So these naked patches of axon are called nodes of Ranvier. And the action potentials, and so you would only find nodes of Ranvier in myelinated axons, right? They're the specific patches in between myelinated sections. So saltatory conduction is just the phenomenon by which action potentials jump from node to node to node. So just going through these different bullet points. Again, no action potentials occur on the myelinated sections of the membrane. They don't have to. Okay? There are no sodium or potassium channels in those sections. The myelin sheath is interrupted periodically by nodes of Ranvier, which are just exposed patches of the axonal membrane. Those are the naked patches I'm talking about. And the membrane current is restricted to the nodes because that's where you find the voltage-gated sodium and potassium channels. We know that you have to have those channels to conduct an action potential, right? And so if you only have those voltage-gated channels in those naked patches, those are the only places you're going to be able to conduct action potentials, right? So the action potentials end up, because these are so well insulated and this depolarization is immediately diffusing to the next patch, you only have action potentials, and the action potentials essentially jump from node to node. So again, this phenomenon is called saltatory conduction. And it greatly increases conduction velocity. So again, on those neurons that mediate fight or flight responses, those neurons involved in the sympathetic nervous system, or those neurons that are involved in spinal reflexes, right? Or like your posture, even though we don't think of that as kind of like a fight or flight, that your posture, you're constantly monitoring your posture, right? You're constantly having to activate neurons in that particular section of your body. And those are myelinated neurons that require very fast conduction velocities. The other thing about this is, if you think about it, if I had to conduct action potentials down this entire axon, if it was completely a naked axon, it didn't have any myelin, imagine how many sodium, potassium, ATPase proteins you'd need on there, right? Because you'd have to constantly be pumping against that driving force to create that driving force. So the fewer patches of membrane that are conducting action potentials, the less ATP has to go towards the sodium, potassium, ATPases, and therefore the less energy is being used to conduct electrical potentials down axons. Does that that's kind of makes sense, right? If you don't have as many action potentials that are being conducted, you don't need so many sodium, potassium, ATPases to be working, and therefore you're saving ATP, right? So that's what this last point is about. The saltatory conduction ends up reducing the metabolic energy that's required to restore intracellular and extracellular ion concentrations. So it's a saver in terms of energy. Many of you have heard of a disease called multiple sclerosis, right? And multiple sclerosis is not the only disease that results in myelin being degraded, but it's the one that's the most studied. So multiple sclerosis sclerosis. This is an autoimmune disorder where have you guys all heard of autoimmune diseases? What's the hallmark of an autoimmune disease? What's going on funky wrong in an autoimmune disease? Yeah, so the body attacks itself, right? The immune system is recognizing yourself as foreign, and it starts mounting antibodies, right, to that particular type of cell, right? Type 1 diabetes, for example, right, is due to the immune system starts attacking the pancreas, and so the pancreas no longer can make insulin, right? That shouldn't happen, right? Anybody who has gluten allergies, so celiac disease, that's an autoimmune disorder. Crohn's disease is an autoimmune disorder of the gastrointestinal tract and other epithelial barriers where suddenly your immune system starts attacking the lining of the gastrointestinal tract. These are things that shouldn't happen, right? And they're very important diseases to study, but we still have major questions about these autoimmune disorders. What triggers the immune system to start attacking itself instead of attacking just foreign molecules? But multiple sclerosis is one of these autoimmune disorders. And in these patients, what happens is that the immune system erroneously starts attacking myelin. So instead of the pancreas or instead of the cells of the gastrointestinal tract, in these particular patients, the immune system is attacking myelin. And what happens over time is that the body is essentially attacking them as foreign invaders. And myelin starts getting disintegrated on these axons. And remember, we need myelination on a lot of our nervous system, particularly those neurons that are involved in fight or flight responses. Motor neurons that are innervating muscle, right, are myelinated. And so in multiple sclerosis patients, a lot of malfunctions can occur. And so people start to usually detect problems when they're lacking some finer motor control, right? Where they are, in their brain, they're saying, okay, I need to grab this coffee mug, but they can't do it because the action potential can't go through saltatory conduction. And it's a degenerative disorder, so it gets worse and worse and worse over time, right? Because you're slowly deteriorating all this myelin. But pretty soon, just mechanistically what's going to happen, right, is that you don't have voltage-gated sodium and potassium channels on these myelinated sections. And if you degrade all this myelin, you might get an action potential up here, but it can't get to that next patch because you've lost your insulation. And so you don't get the neurotransmitter released from the axon terminal, and therefore you don't get the action on the end, right? You don't get the muscle contraction, for example. So multiple sclerosis is, any time that you have a disease that can describe kind of pathophysiology, it helps us understand normal function, right? And so it helps us understand how important myelin is when we see what happens when it gets destroyed. I showed you this picture before. This is an electron micrograph of a myelinated axon. So again, here you see the cytoplasm inside the axon itself, and all these are the organelles, vesicles, and other organelles that are inside. Here's the plasma membrane of the axon, and then around here, these concentric rings, that's the myelin sheath, okay? And so then you can see in this just sort of artist rendition, the axon is shown in yellow and the myelin sheath is shown in purple, right? And so this also shows you in an artist rendition the myelinated sections, what I sort of tried to depict on the board, but this obviously looks a lot better. And you see the nodes of Ranvier. You have these sections of naked axon, right? And that's where the action potentials are conducted. You don't have to have action potentials in these myelinated sections, and in fact you wouldn't because there are no sodium and potassium voltage-gated channels. This is just kind of a circuitry diagram of the phenomenon of saltatory conduction. Here are these patches of myelinated axons where you see the purple, and you can see that you don't have sodium and potassium channels on those myelinated sections. They're only in these naked nodes of Ranvier, and you can see that you literally have an action potential that's jumping from node to node because of the quick spread of depolarization on these myelinated sections. Are there any questions about myelination or action potential propagation? Because what we're going to talk about now is how an action potential ultimately gets to start. How do you reach that threshold? And we're also going to talk about the end, right? What happens when you get an action potential down to the axon terminal? What happens at a chemical synapse? Okay. So I think you guys all know basically that neurons that communicate via chemical synapses. Remember, we also have neurons that communicate via electrical synapses. But those are in the minority, although they play an important role in the nervous system. But we're talking mostly about chemical synapses, right? And we know that the synaptic cleft is anywhere from 20 to 30 nanometers across, right? It's bigger than the electrical synapse. And I think everybody knows the basics, that this would be the synapse, this would be called the presynaptic neuron, right? And so this would be called the postsynaptic. But I'm just going to keep this neuron on the board. Wouldn't this be the presynaptic neuron for this neuron? So true or false, a presynaptic neuron can also be a postsynaptic neuron. True, right? So, but we use these terms to define what's going on in the case of two neurons, right? So you guys know the basics, that you get these particular electrical signals that happen in the cell body. Those electrical signals are summated, and if there is a threshold potential that's surpassed, you initiate an action potential. So we've spent a lot of time talking about what happens here, right? We've spent less time talking about those magical electrical signals that happen here, and we've spent very little time talking about what happens at the axon terminal. So that's what we're going to talk about now. So what you see on the board is something called a postsynaptic potential. What that means is if we're talking about this synapse, right, this action potential is going to come down to the axon terminal, and we'll talk about the magic that happens here. But when that depolarization wave comes down to the axon terminal, that opens up some voltage-gated calcium channels. So the axon terminal doesn't have the same types of channels that the axon itself has. The axon terminal has voltage-gated calcium channels. Stand up. Oh! Sorry. Voltage-gated calcium channels. Now, calcium is much higher in concentration on the outside of the cell than on the inside of the cell. And so if the action potential, that wave of depolarization, opens up those calcium channels, calcium is going to rush right in to the axon terminal. And you guys now know from our example of the GQ, G protein coupled receptor pathway, that calcium is a major second messenger, right? And calcium is hugely important as a signal for biological responses. So calcium suddenly now is high in concentration on the inside of the axon terminal in the cytoplasm, and that causes neurotransmitters to be released into the synapse. We'll talk about the process by which that occurs, how calcium activates neurotransmitter vesicles to fuse with the membrane. But for now, let's just say, okay, action potential is propagated down the axon. That causes calcium channels to open. Calcium rushes in, and neurotransmitter is dumped out into the chemical synapse, right? And that we'll go through all the classes of neurotransmitters, but you guys so far have heard of norepinephrine, and you've heard of acetylcholine, and I'm sure you've heard of serotonin and dopamine and glutamate, right? You've heard of all these different types of neurotransmitters. So now, if I am a neurotransmitter that's been dumped into the synapse, what does the postsynaptic neuron have to have on it in order to respond to that particular neurotransmitter? A receptor, right, for that particular neurotransmitter. In the case that there is a hydrophobic neurotransmitter, like nitric oxide or carbon monoxide, if you're exposed to carbon monoxide, those can actually pass right through membranes, so you wouldn't need a membrane receptor for those, right? But most of the ones we're going to talk about are neurotransmitters that would stimulate membrane receptors, right? Okay. So these membrane receptors will be cause some sort of biological response, right? Some of them are ion channel linked receptors. We have 3 types of membrane receptors, right, everybody? You guys remember that? We have ion channel linked receptors, which a lot of these happen to be. We have enzyme linked or enzyme receptors, right? And we have our G protein coupled receptors, right? So we know that these neurotransmitter receptors there's nothing special about them. They're the same types of receptors that you guys have already memorized the pathways for, right? Okay, so the neurotransmitters bind to their receptors, and we'll talk about the specifics of some of these. But the most important thing to understand is that the biological response in this case is an electrical change. So what's happened is that a chemical signal is then transduced as an electrical signal. And these are called postsynaptic potentials. You guys have heard of potentials, you've heard of resting membrane potential, you've heard of action potential. All a potential is a difference in voltage across the membrane, right? And so something called a postsynaptic potential would just be an electrical difference across the membrane that's in the postsynaptic neuron. So let the definitions kind of work for you. Okay. We also have talked before about graded, where you can increase or decrease, kind of like the dial of a volume dial, right? Versus all or nothing. Like in action potential, it's not going to happen if it's a sub-threshold stimulus. But the minute that you have a super-threshold stimulus, you're going to get that same magnitude response, right? That's not the case with postsynaptic potentials that happen in the cell body. Postsynaptic potentials are graded. That means, let's just say that binding this neurotransmitter to this receptor, let's say that those opened up a conductance to sodium. Say those were ion channel linked receptors, and once the neurotransmitter bound the receptor, you opened up that was actually a channel, and it was a sodium channel. So now suddenly you've let some sodium in. What is that, a depolarization or a hyperpolarization? If sodium rushes in, what is that? Depolarization, right? And so that, if we were to draw that on, on a graph, that would be kind of going more positive, right? Alternatively, if these were like chloride channels, what charge is on a chloride ion? Negative. If these were chloride channels, and chloride happens to be more abundant on the outside of the membrane too. If these were chloride, if these receptors opened up a conductance to chloride, what would happen on the inside of the cell? A depolarization or a hyperpolarization? A hyper, right? And so in that case, I'm going to draw a different neuron for that. If chloride ions came in, that's going to actually hyperpolarize the cell, right? These are small electrical potentials that are going to be elicited by neurotransmitters binding their receptors. So let's just take our depolarization case. Maybe I had one action potential come down this presynaptic neuron, so I had a little bit of neurotransmitter in the synapse, and I had a little bit of neurotransmitter binding its receptors. I had a little bit of depolarization, but it was tiny and it wasn't enough to be super threshold. So if it's not enough to be super threshold, am I ever going to get an action potential? Nope. And that decision is made when you start getting voltage-gated sodium and voltage-gated potassium channels, right? And those are on the axon. So if I just get a little depolarization, that's great and everything, but that's not going to cause those voltage-gated sodium channels to open, and therefore you're not going to get an action potential, and the signal's going to stop, right? Same thing holds true for, what if I have a neurotransmitter binding to chloride channel receptors, and I cause a hyperpolarization? Am I going to get voltage-gated sodium channels to open if I hyperpolarize the cell? No, remember those are depolarization activated channels, right? So I'm going to get way further away from the opportunity to have an action potential, right? Okay, what if I have a high frequency of action potentials coming down this axon, this presynaptic neuron? I get a ton of neurotransmitter dumped out into the synapse. I get a bunch of these receptors bound with neurotransmitter, and I get a much bigger depolarization that is super threshold. Am I then going to be able to get an action potential? Yeah, and you'll have an action potential, and it'll go all the way down this axon because it's all or nothing, right? So these potentials, these postsynaptic potentials, are graded, right? They are not all or nothing. They are sometimes small, sometimes bigger, sometimes much bigger, and unless they reach threshold, that threshold potential for those sodium channels to open for the action potential, you're not going to conduct that electrical signal. The other thing that you need to keep in mind is that more than one neurotransmitter can be in a synapse at one time. So let me draw another picture over here. So, this would be an example of what's called convergence, where you have many neurons that are kind of synapsing on one. So, you guys can see that this is still a synapse, right? And this guy is presynaptic to this one, and this guy is presynaptic to this one. This is the postsynaptic neuron, right? But neuron 1, let's say it releases acetylcholine, right? Let's say this is a cholinergic neuron. All cholinergic neurons release acetylcholine. Just remember that choline thing. All cholinergic neurons release acetylcholine. In fact, here's another fact of the day. Neurons can only make and release one type of neurotransmitter. Everybody catch that? That kind of makes it easy to remember. One neuron, it can only ever make one type of neurotransmitter. So if I'm a cholinergic neuron, that's it. I make acetylcholine. That's all I do. I mean, obviously, I conduct electrical signals and all that stuff, but I only make acetylcholine. If I'm a serotonergic neuron, I only make serotonin. If I'm a dopaminergic neuron, I only make dopamine. If I'm a nitric oxide neuron, I only make nitric oxide, right? So, true or false? One neuron only makes one type of neurotransmitter. True. But isn't it true in this case that neuron 1 could make one type of neurotransmitter and neuron 2 could make another?
Yeah.
Okay, so neuron 1 could make a cholinergic, and let's say neuron 1 could make acetylcholine. And let's say neuron 2 is a GABAergic. Have you guys heard of GABA? Gamma-aminobutyric acid. So GABA, correct me if I'm wrong, neuroscience students, but GABA is inhibitory, no? It's inhibitory, I'm thinking. So let's just say this is GABA. And a GABA, so these are actually called GABAergic neurons. I don't make these things up, I'm serious. So GABAergic neurons, okay? So the acetylcholine is an excitatory neurotransmitter. Simply because its receptors, you guys remember the nicotinic acetylcholine receptor? You also have one called the muscarinic acetylcholine receptor. Anyway, its receptors are excitatory. That means that they are going to open up conductances that are depolarization events. So, let's say you have a little bit of acetylcholine, just a little bit, like this guy had maybe one action potential that went down the axon, and you had a little bit of acetylcholine, so you had you had a small postsynaptic potential. That is called, by the way, an excitatory postsynaptic potential, because you're moving the membrane potential towards threshold, okay? So that's called an EPSP, excitatory postsynaptic potential. But what if at the same time GABA was released, you had an action potential coming down this guy, and GABA was released, and it opened up conductances to chloride, and suddenly you get a bunch of negative charges. So if you were to trace the electrical impulse there, it's going to look like that. What do you think these are called? Not excitatory postsynaptic potentials, but inhibitory postsynaptic potentials. IPSPs. Okay, so that would be an IPSP. Well, in terms of this axon reaching threshold, so that an action potential will go down its axon, what's the issue? What do you think is going to happen?
Nothing.
It's going to flatline, right? If there's the same amount of sodium channels and chloride channels that open, aren't they just going to sort of cancel each other out? And your membrane potential is essentially going to stay at -70, right? hyperpolarize and way unlikely that you're going to have an action potential, right? What if the opposite was true? What if you had a lot of firing on this one and very little on this one? You might hit threshold because you're going to get a lot of sodium rushing in, and the EPSP is going to be much bigger, and it's going to go towards a super threshold stimulus. And if the threshold is high, if the potential, if the EPSP is high enough, you're going to kick off an action potential, right? Because those voltage-gated sodium channels at the axon hillock, which is right, it's kind of the neck of the axon here. That's where the sodium and potassium channels start residing, right? Those sodium channels, they ain't gonna open unless you get that threshold potential, right? And so the EPSP, if it dominates and if it's super threshold in that particular neuron, you're gonna get an action potential, right? So let me make sure that I've gone through these points. So remember, postsynaptic potentials, whether inhibitory or whether excitatory, are graded. And graded potentials that have resulted from channels opening are summed. They're summated. Right? And so it gets pretty complicated. I mean, think of how many millions, billions of neurons you have in your brain and how complicated these synapses are. Where you have, I mean, I'm giving you the simplest case here, where you have two converging on one. I mean, you have hundreds converging on one another and then diverging because one neuron can have multiple axon terminals. Even though it releases the same neurotransmitter, it can actually synapse on many, many, many other neurons. So this is going to be an interplay between inhibitory and excitatory neurotransmitters that are released and how frequent the action potentials are in the presynaptic neurons, right? Yeah?
And this whole time, you have the sodium-potassium pumps trying to get back to -7.
All the time. All the time. They're just pumping away.
Okay.
But the closer you get to -70, the less they have, the less they have to work, right? So that's why more myelin, fewer sodium, potassium, voltage-gated channels, less sodium, potassium, ATP is required, right? But they're always going. Yep. So postsynaptic potentials, like I said, can be excitatory or inhibitory, depending on the neurotransmitter or modulator that acts on the neuron, right? So the electrical response is completely dependent on the neurotransmitter that was released by the presynaptic neuron, right? So imagine a more complicated situation where, say, you had 10 presynaptic neurons, and this postsynaptic neuron, in order to respond to their neurotransmitters, would have to have the receptors, right? So think about this. I know this is kind of going by lightning fast, but true or false, a postsynaptic neuron only has one type of neurotransmitter receptor.
False.
That's false, right? Even though this guy can only release one type of neurotransmitter, it can respond to a lot of different types, right? So I might have receptors to 5 different types of neurotransmitters, but I myself can only make one type of neurotransmitter. I am a cholinergic neuron, or I am an adrenergic, which is adrenaline, epinephrine, right? Adrenergic neuron, or I am a serotonergic neuron, etc. Okay, so did you have a question? Okay. So this is just showing you the difference between an excitatory postsynaptic potential and a regular old action potential, right? These should look to you like action potentials, right? This is an action potential, this is an action potential, right? So what we're showing here is, if this was the action potential that came down the presynaptic neuron, if this was an excitatory neurotransmitter, this is the type of depolarization event that you get. And you can see it's time. I mean, this is 1 millivolt of a change. So that would be one action potential coming down a presynaptic neuron would release enough neurotransmitter to elicit that small of a change. And if it was an inhibitory neurotransmitter, again, that small of a change. So it's going to be higher frequency of action potentials is going to cause a much bigger depolarization when those are summated, and vice versa in this case, right? And those two are going to be summated at the axon hillock. So if say the neuron had a threshold potential of minus 55 millivolts, right, where those voltage-gated sodium channels aren't going to open unless you're at minus 55. So you can see that, one EPSP is not going to do it. So if you get one action potential coming down the axon, typically that's not going to be enough neurotransmitter to cause a suprathreshold stimulus. I say typically because when we talk about skeletal muscle physiology, one action potential down a motor neuron is enough to release enough acetylcholine to elicit a twitch, a muscle twitch. So, but in neuron-to-neuron communication, one EPSP typically is not enough. One EPSP elicited by one action potential in the presynaptic neuron is typically not enough. So this is just showing you, if I have EPSPs, it's bringing me closer to the threshold, but not enough unless I summate a bunch of them and I'll get an EPSP that's big enough. An IPSP is actually pulling you away from the threshold. So it's inhibiting the chance, or it's making the chance less probable that you're going to get an action potential. So this is just kind of a review of what we've talked about thus far. EPSP or IPSP totally depends on the channels that are opened by the neurotransmitter or the signal. Depends on the biological response elicited by the neurotransmitter and the receptor. So, like we've talked about acetylcholine a lot, and do you guys remember that nicotinic acetylcholine receptors are actually cation channels? That they open up a conductance to both sodium and potassium, but the sodium driving force is way, way stronger, and so sodium rushes in, right? Exactly. So this, if you want to in your notes, make a note that this is like the nicotinic acetylcholine receptor, NACH, right? So if this was acetylcholine and I bound to my receptor, that would open up this cation channel, and that would cause sodium to rush in and a little potassium to rush out, but the driving force, as you guys know, is way higher for sodium. And so you would get an EPSP as a result. Some neurotransmitters activate calcium ion channels. And if you open up the conductance to calcium, you guys should know by now that calcium is much higher in abundance on the outside of the cell than on the inside, and so you're going to have calcium rushing in. Calcium is a cation, right? It's a divalent cation, so you're going to increase positive charges in the cell, and that's going to cause an EPSP. Some neurotransmitters are going to open up chloride channels, and that's going to cause an IPSP, right? So whether it's an EPSP or IPSP is completely going to depend on the neurotransmitter that stimulates its receptor. So this is exactly what I drew on the board, but you have it in note form here. If I had an excitatory neuron versus an inhibitory neuron, and this one was, my example here was a sodium channel, but here's a calcium channel. So if my neurotransmitter bound to a receptor that was a calcium ion channel, that would cause an EPSP. This neurotransmitter would cause chloride channels to open, causing an IPSP, and the graded postsynaptic potentials will be summated. And if the sum potential is greater than the threshold, if it's a suprathreshold EPSP, then an action potential will be initiated at the axon hillock. But if they just cancel each other out, then it won't be suprathreshold, so you won't get an action potential. So after you get we have plenty of time here. So after we get the action potential sent down the axon, the major importance of calcium is apparent. So don't get this calcium mixed up with the calcium that we're talking about next. So I should stop for a second there and say, does anybody have any other questions about postsynaptic potentials? Postsynaptic potentials are graded, they're summated, and they are completely due to the opening or closing of ion channels due to neurotransmitters that are binding the receptors. Okay? And you're not going to get an action potential down that postsynaptic neuron unless you reach that threshold potential, right? So now what we're going to talk about, now we have the axon part done. We understand what causes an action potential. Now we understand what happens in the cell body that's actually going to start an action potential going down the axon. We understand how myelination plays a role in conduction velocity and how important that is. So now we're going to focus a little bit of time talking about the end, the last thing that happens. So we already talked about on this particular neuron, when the action potential travels down the axon terminal membrane does no longer have voltage-gated potassium and sodium channels, rather it has voltage-gated calcium channels on it. So this part of the membrane is flush with sodium and potassium voltage-gated ion channels, except for myelinated regions. But this membrane is rich in voltage-gated calcium channels. And they're voltage-gated and they open in response to depolarization. So when that wave of action potential comes down the axon, remember that sodium channels are opening and sodium ions are rushing into the cell. That wave of positive charges, once you get to the end, is going to open up calcium channels. They open up there in response to depolarization. So after the action potential is sent down the axon, voltage-gated calcium channels that are embedded in the axon terminal open. And you guys now know the direction of the driving force on calcium is always going to be into the cell, right? Because the calcium ion concentration in the cytoplasm is very low, but it's very high on the outside, right? So if I open up a calcium ion channel, calcium's going to rush in. This is the part where the electrical signal of the action potential now turns back into chemical. Because it's not the calcium's charges in this particular case that's important. It's the calcium ion itself. It's the chemical calcium that's important. Because what calcium is going to do is it's the major trigger for exocytosis of neurotransmitter-containing vesicles. So what I'm going to do in the last couple minutes is just draw a huge axon terminal. So here's the axon, and here's the axon terminal. Does everyone understand that this is the axon? Here comes the action potential, right? So these are calcium channels. And what we haven't really talked about yet is that waiting, as if they're, runners that are ready for the gun to go off in a race, are vesicles that are filled with that particular neuron's neurotransmitters. So again, if I'm a cholinergic neuron, what's going to be stored up in my vesicles if I'm a cholinergic neuron? Acetylcholine, right? If I'm a adrenergic neuron, what's going to be stored up in my vesicles? Adrenaline, adrenergic. What is it going to be? Adrenaline, or epinephrine we call it now, right? Epinephrine. So these are where your neurotransmitters are stored in vesicles. And we'll talk about this. I'm going to finish this lecture this afternoon before we start our neuro exercise. But these vesicles are tethered. If they weren't, they would just be bopping around all over the cytoplasm. But they're tethered, they're anchored right by the plasma membrane, and they're waiting for that calcium signal. And when calcium floods the axon terminal, that's the stimulus for these vesicles to fuse with the plasma membrane. And just like a bubble fuses with another bubble, they're going to dump out their neurotransmitter, right? They just fuse, and that's called exocytosis, right? Did everyone kind of see my little sleight of hand there? Right? There's just a fusion of these vesicles with the membrane. But this can't happen unless these vesicles are released from their tether. And so now the neurotransmitter is outside in the synapse, right? And now it combined to the postsynaptic neuron, and so on and so forth. Then you get an EPSP or an IPSP, and then you get if you reach threshold, you get an action potential, and so on and so forth. But calcium is the major trigger. So you get the series of actions are an action potential, travels to the axon terminal, voltage-gated calcium channels open, calcium rushes into the cytoplasm. Calcium serves as a trigger for exocytosis of neurotransmitter-containing vesicles. Neurotransmitter is released into the synapse, and then you can get activation or inactivation of the postsynaptic neuron. So we'll talk this afternoon about how neurotransmitters are regulated in the synapse and about some drugs that can affect neurotransmitter concentrations in synapses.